Hello all, my name is Rashni Pinwala and I did my term paper on the techniques for improving coffee production, specifically through CRISPR Cas9 transformation and then further propagation through somatic embryogenesis. So let me begin by explaining why the improvement of coffee production is so critical. So coffee has been proven to be highly sensitive to climate change, and because coffee plantations have a lifespan of about 30 years, the likely effects of future climates are already a concern. Most coffee in general is produced from two different species. Most well known and used around the world is Arabica coffee, which accounts for 70% of global production. And then you have Robusta coffee, otherwise known as Cafe canephora, which is the main topic of this presentation, which accounts for 30% of global production. But overall, it's known that Robusta is generally more heat tolerant, which is really important as climate change begins to go further. So climate change has already been forecast to reduce productivity of Arabica coffee, and forward-looking research on adaptation is therefore in high demand across the entire supply chain. And you can see in this figure right here, in a study done by Bunn et al., it suggests that higher temperatures may reduce yields of Cafe Arabica, while canephora potential areas grow at an overall gain as climate change continues to rise over the next few decades, which you can see here indicated by the large amount of green, while the red in the Arabica section kind of indicates the loss that's going to be happening for that specific variety. And Arabica also suffers from greater pest attack by the coffee berry borers, as depicted by this chart, and because of that, they're a highly threatened species. And although canephora can sustain higher temperatures and is less likely to be harmed by the coffee berry borer, there's a higher want for Coffea Arabica, and most literature for transformation is corresponding with Arabica because of that. So it's really important that we begin to kind of shift our mindset to focus on transformation for canephora over Arabica just because of its future potential as climate change begins to rise. And it's also really important to remember the livelihoods of millions of people depend on coffee. And so it's not just of agronomic importance, it also is of socioeconomic importance, and we need to be able to do further research that will help us develop varieties that are going to withstand the changes that are coming with the climate. So current techniques that are kind of known for coffee right now, the main technique overall is the pedigree breeding method to produce F1 hybrids, and that's to produce most elite varieties of coffee. And then further on with that, the use of somatic embryogenesis was introduced as an effective means of propagation for those new hybrids. And what's really important to kind of establish here is that there's major limitations that are associated with conventional breedings, such as how long it takes to, how long the generation time is for a new coffee tree, or the cost of field trials, or the lack of accuracy of the breeding process. And it's really critical that we're starting to build that genetic resistance of those elite varieties over the next few years. So what was kind of introduced into this was a new form of transformation, which is through agrobacterium, to introduce genes conferring resistance and tolerance to biotic and abiotic stressors. And the major point here is that you have new techniques that can be established and used, and specifically we're going to be looking at CRISPR-Cas9 as a way to help introduce new traits to already established elite coffee genotypes. So the paper that I focused on as my primary research article is this one right here that was done by Bretler and Deschamps of the French Agricultural Research Center. And it's kind of talking about the ability to domesticate Coffea canephora at a higher rate as Arabica is starting to decline because of climate change. So the main hypothesis of this paper was that genome editing by the CRISPR-Cas9 method is an efficient and reliable way of knocking out genes of agronomic interest in the coffee tree. Developing the CRISPR-Cas9 system in coffee would greatly accelerate research and the breeding of new varieties, and thus shortening the overall time for breeding, which is very, very attractive for anyone who wants to generate a new cultivar with improved agronomic traits. So the way they did this was first by using a CRIP program, which stands for Coffee Guide RNA Identification Program. And the authors used the CRIP program to identify all the possible CRISPR guide sequences, starting with the G and followed by their PAM sequence of interest, which we'll kind of talk about in the next slide. And this web tool uses an algorithm that searches for the potential off-target sites for each CRISPR guide sequence identified. So this is, I don't know if you can see my mouse, but the TGG and the AGG and the CGG is that PAM sequence that I was referring to. And so in order to verify whether the CRISPR-Cas9 system could effectively direct gene-specific editing in the Robusta coffee genome, they selected CCPDS, which is the phytoene desaturase gene in the coffee genome hub, as the target of the Cas9 endonuclease. And this gene was selected since it's already been extensively studied to investigate gene silencing in other plants, such as poplar and apple trees. And they know that the mutants of this PDS genes produce an albino phenotype, so whenever they're trying to figure out if it's lost during this transformation process, we're able to see it through the albino phenotype. So they selected 3 sequences with their PAM site in the 3' regions of the CCPDS locus, and they as unique gRNA complementary sites with potential off targets. Those 3 were the PDS3, the PDS5, and the PDS14. And these gRNAs were then assembled using PCR and cloned with Cas9 endonucleases, coding for this binary vector that's specific to each of these. You can see right here that I'm pointing to. And using these 3 vectors in agrobacteria-mediated transformation of coffee embryogenic callus, Cas9 and each gRNA could be simultaneously expressed in the coffee cells. So the actual results of this showed that the independent transgenic lines of each exon binary vector produced mutations in only the PDS14 when the PDS14 was targeted. So 30.4% of the plants regenerated with binary vector for the PDS14 displayed mutation in this PDS alleles, with 7.6 corresponding to biallelic homozygous mutation. They confirmed that no SNPs, nucleotide polymorphisms, were detected at the target sites, although 2 SNPs upstream of the target sites were found. Based on this information, it was possible for them to distinguish that 2 copies of the PDS gene in the Coffea canephora genome, and that the CRISPR-Cas9 could edit each of those 2 alleles independently of each other. And so for the transformation and regeneration step to kind of understand what's happening here, as stated previously, mutations could be detected at the desired target only when the last exon, the PDS14, was targeted, and it was seen that 30% were actually mutated. None of those plants, however, displayed a full albino phenotype, as it should have occurred with PDS mutants, as we stated before, but they did display a range of severe mutant phenotypes, as you can see right here. These second pictures, and then this one as well, were all the mutants. And then the 9 different mutations identified in the DNA by these specific vectors corresponded to 7 different mutations in the amino sequence of the PDS protein. No mutant plant could be detected with exons 3 and 5 of this PDS gene, and were targeted despite the sequencing of 150 transgenic plants. Because of this, a new binary vector bearing 2 gRNA simultaneously targeting those 2, 3, and 5 exons, and a Cas9 coding sequence optimized for plant expression was constructed by the authors. And the results showed that embryogenic calli formed with the construct could not regenerate transgenic somatic embryos, while the calli transformed with the second binary vector, with the 2 binary vector without gRNA systematically regenerated numerous transgenic somatic embryos showing a normal phenotype. And that's displayed in this item that's circled right here. And what that's really saying is that we were able to see the gene being transformed. And so using our first binary vector bearing a single gRNA mutation, mutation efficiency could reach 30%. And most important, the generation of homozygous knockout mutations in the regenerated plantlets opened up the way for further coffee molecular breeding programs with their perspective of shortening the time needed to create new cultivars. And that's a significant decrease, where it's going from maybe 25 to 30 years of a large breeding program to potentially 6 years if you're able to identify the gene of interest very quickly. And this is definitely compared to classic pedigree selection. You're able to target genome editing. You're able to target the genome, and it raises no barriers when it's coming to different coffee species. Like we had mentioned earlier, you might have difficulty with ploidy levels of other types of species, whereas in this one, it can be applied to all the different types of coffee genomes, whether it's canephora or arabica or another one. And there is also potential for CRISPR-Cas9 to be applied to other trees as well, and perennial species like we had mentioned earlier, like the poplar and apple, since they also have the same PDS gene function. And in conclusion, a couple of points that I'd like to reiterate is the fact that as climate change continues to be a problem for coffee yields and will likely cause coffee production to drop, it's really important that we have a new mechanism to be able to develop new cultivars and varieties at a faster rate. And the results defined in this primary research article demonstrated that the genome editing by CRISPR-Cas9 method is an efficient and reliable way of knocking out agronomically interesting genes in the coffee tree. And they also showed that the use of somatic embryogenesis is a way of regenerating plants that are genome edited by CRISPR-Cas9 technology, which is also important if you want to continue to propagate those plants and have them producing an actual yield that can be used around the world. And finally, this research with genome editing technology could allow coffee to rapidly introduce new traits in already known elite genotypes that could help with the steady need for high quality coffee production as consumption continues to rise through the next few decades, specifically because we have that issue with climate change effects going on. And these are a few of my references. Thank you.